Alright, so chapter 14 now, all about RNA and RNA processing of messenger RNAs. Talks a little bit about tRNAs, ribosomal RNAs, microRNAs, things like that. Lots of things about RNA. And I'm going to expand on the answers that the book gives. They're not usually all that informative, which hopefully makes it worthwhile of me doing this. So what are some characteristics of introns? I mean, that's a pretty vague question. The book talks about that introns are intervening sequences that are in the middle of genes, protein coding genes. They don't really say all that much about it. So what are some characteristics? Only in eukaryotes. There's various types of introns. The main ones we'll talk about are those that are involved in protein coding genes. So we know a protein coding gene looks like that. And there can be any number of introns and exons. And so I usually write it with just 3 exons. And so then the introns, so we get transcription. And then the introns get spliced out, and the exons get spliced together. And if we focus in on one of these introns, when I think of what are the characteristics of introns, I want to know what is it about this that makes it an intron. And we'll go over the mechanism of splicing, but as we've said before, DNA and genetics and DNA, RNA, protein flow of information is all about information. And so there has to be some information in here that makes this an intron. So why, when I draw this little line in these boxes, how does the cell know that there's an intron there or an exon there? And so there are sequences that are part of the intron that define it as an intron. Now there are some other sequences in the exon that overlap here that are also help the cell know that that's an intron. But what's always the case is that there's a GU at the 5' end. So this is the 5' end of the RNA. This is the 3' end. And so this is the 5' splice site. And this is the 3' splice site. This is also called the splice donor. And this is called the splice acceptor. And this is called the branch point. And when we go over the mechanism of how the RNA gets spliced out, we'll show you what each of those things does. But those are the sequence determinants and that cause this to be an intron. So it'll get spliced there, it'll get spliced there. And after it gets spliced, the 2 blue things get spliced together, and the intron gets spliced out and goes away, and it forms this little branch point here. So we'll come back to that. And I'm not going to worry. There's group 1 introns, group 2 introns, all that kind of stuff. Not too worried about that. In my opinion, that's just a little bit too much detail on exactly what introns are. And so this is an intron that's in a pre-mRNA. All right. And what are the 3 principal elements in mRNA sequences in bacterial cells? There's not a whole lot going on with RNA in bacterial cells, at least at this level that I know of. So here's a bacterial gene. And These are not exons, though. These are individual open reading frames. And so when this gets transcribed, it gets transcribed into a polycistronic RNA. And so it's going to, the RNA is going to start right at the transcription start site. And so when they talk about the different characteristics, what we have is we can think of all of this as a coding region. This happens to contain 3 open reading frames, so it codes for 3 proteins. And this is the 5' end of the RNA. This is the 3' end of the RNA. And this would be the first start codon. And then there'd be another. But each of these open reading frames has its own start and its own stop. And there's 3 different stop codons. There's only 1 start codon. There's 3 different start codons, so I'll use a different one there. So this region up here doesn't get translated, and so that's called the 5' untranslated region. So that's the 5' UTR, and that is the 3' UTR. So the official answer in the book is the 5' untranslated region, the protein coding region, and the 3' untranslated region. But in here, there's a sequence when we talk about translation where the ribosome binds. So this is called a ribosome binding site. Or the RBS. And it is also called the Shine-Dalgarno sequence. Alright, so what is the Shine-Dalgarno sequence? That's the next question. If you have a prokaryotic RNA, Shine-Dalgarno is only in prokaryotes. Prokaryotes, and it's a ribosome binding site. And so it is positioned, or its location is upstream of the transcription start site, and it's often written as just the SD sequence, just named after the 2 people that discovered it. It's called the Shine-Dalgarno sequence. And its function is that's where the ribosome binds. If we have a start codon down here, and we make our RNA, so it would be ATG and DNA, AUG and RNA. When the ribosome comes in to translate this, it's going to lock on to that Shine-Dalgarno sequence and then move along to the start codon and start translating. So it's a ribosome binding site, which is exactly what it sounds like. It's a site where the ribosome binds. What's the 5' cap? The 5' cap is a cap that goes on the 5' end. But this is only in eukaryotes. So this is eukaryotic. And I always just sort of draw it as a little asterisk saying that we've got this cap there, and we'd have our exons spliced together, and we'd have a poly A tail, only in eukaryotes. And this cap happens to be a 7-methyl guanosine cap. So it's actually a nucleotide. So if we have a guanosine, which looks something like that. I don't know exactly where it is, but somewhere on there, a methyl group gets added, and then this would be a guanosine. That would be G. We got our phosphates. It actually gets added kind of in a weird way. This 5' phosphate gets added, but we don't really know the need to know the structure. You might need to know 7-methyl G is a cap that gets added onto the end of the RNA. And its function in eukaryotes is for the ribosome binds onto that. So that's a difference between prokaryotic and eukaryotic translation, is the ribosome binds onto that cap and then scans down to find the AUG, and then it starts to translate. And we'll talk more about translating in chapter 15. How is the polytail added? You got a gene. So you'll notice I draw this diagram a lot. So you should get used to drawing this. You've got some exons and introns, transcribe, and then we're going to splice out the introns and put these guys together. Let me erase the whole thing. So poly A tail is part of RNA splicing or processing. It's one of the processing steps. And so the processing steps are one, not necessarily in this order, but they all happen at some point. Adding the 5' cap, and 3, adding the poly A tail. And so this cell, as it's making this RNA, there's going to be this signal here called the poly A addition signal. Very good. Then when we transcribe that, it becomes AA, UA, A. Put those in there twice for some reason. But that's not where the poly A tail gets added. There's a protein that binds that, and it's called the. So this is in the 3' UTR. And this is the 5' UTR. So both bacteria and eukaryotes have a 5' UTR and a 3' UTR. That's untranslated region. And there's a protein complex that recognizes this and then cuts. No, it doesn't cut it there. It cuts it down here, and then it actually chews away a little bit, and then it adds poly A tail. So it's going to add the poly A tail maybe right there. So it gets rid of some of this, and then adds the tail. So this signal, this protein actually says, okay, cut there, and then there's other proteins involved that come along and bind and cut the RNA and add the tail. And the function of this tail, once you have this tail, it adds, it makes the RNA more stable. So maybe there's RNAs that don't get completely transcribed or something. If you take away that poly A tail, the RNA will get chewed up and degraded. So then you've got your exon spliced together. You got your 5' cap, your UTR, and your 3' UTR. So that's that. And then what is it? So what's the spliceosome? The spliceosome is the complex that splices out the introns. So if we've got our intron, if we focus in on an intron, there's a series of proteins that bind here, and then another one binds here, and then another one binds here, and these proteins are made up of, so what makes up the spliceosome are things called SNRPs. And SNRPs are made up of RNA and protein. The RNAs are called small nuclear RNAs, because splicing happens in the nucleus. So here's a nucleus. And mRNA gets made, and it has to get spliced, but eventually the mRNA makes it to the cytoplasm, where it's going to get translated. But the RNAs that are involved in SNRPs are these small RNAs that don't go out of the nucleus, and they're called small nuclear RNA. So SNRPs are made up of small nuclear RNAs called snRNAs, and then various proteins, and then together this makes a SNRP. And all of these SNRPs together, then some other proteins will be involved, and there might be some, these have some little RNAs in there, and together this whole thing is called the spliceosome. So what makes up the spliceosome? SNRPs. What makes up SNRPs? Small nuclear RNA and protein. Because they're made up of RNA and protein, they're called ribonucleo, or ribonuclear sometimes, but I think it's ribonucleo protein. That's called an RNP. So it's really a small nuclear, so it's abbreviated small nuclear ribonucleoprotein is a SNRP. And they make up the SNRP, which makes up the spliceosome, which splices out the intron. Alright, so let's focus in on one intron. And we'll give you the steps without the SNRPs, and then we'll throw the SNRPs in there. So all of these SNRPs, all of these steps are catalyzed by SNRPs. But the first step is to cut the RNA at this point. You know, this kind of happens in one step. So we're going to cut this, and then this is going to form a bond here. So we'll draw it like this, and this G and this A form a bond, and then this X, and so this is the exon right here, and so it cuts that and forms that bond, and this is all within the context of the spliceosome. Then the next step is these exons go together, and this lariat in the middle, so this part is released. call that the lariat. And then this eventually just gets degraded. Now there are some functions for these things now, they think, but we're just talking about the basic mechanism at this point. Alright, so that gets degraded. Now the one thing that's interesting is I always want to point out, because this looks a little bit confusing, we got an AG bond right here. So we got this G and this A are forming a bond, and it's a covalent bond. So this is not an AG base pair, they're not base pairing. They're actually forming a phosphodiester bond. But it's between the 2' hydroxyl of the G, no, of the A. So it's the 5' part of the G and the 2' part of the A, and they form a bond. So that's where that one of the places where that 2' hydroxyl. So if you think about RNA structure, it has this OH. Now we're used to seeing a 3' OH. That 3' OH can form your normal phosphodiester bond, but now we're forming a phosphodiester bond using our 2' OH. So it's a 2', 5' phosphodiester bond. It's very specific for that particular reaction. Now splicing can give you different outcomes from the same gene. So we have a gene, and so let's say this is exon 1, exon 2, exon 3. And we can represent splicing like this. We can say that we're going to join those two things together, and we're going to join those two things together. In each case, we're going to use that same mechanism that we just showed, but we just splice these together and get 1, 2, 3. But we could also splice this differently. So this is a GU, and this is going to be an AG down here. Maybe we skip that middle intron and we just splice from here and we go all the way to there. In that case, we'd have 1 and 3, and we'd skip that exon. So that is called alternative splicing. And we could get different outcomes from that. So we're cutting out the middle bit of that gene. If we're getting rid of this, that might have some interesting protein information, and it would give us a different protein for version 1 over version 2. And so this is one alternative RNA processing pathway, alternative splicing. And so we have the same gene, but now we've got two different proteins, or two different RNAs, and those can give rise to two different proteins. So this, if we translate that, we'll get a protein of a particular length, but let's say we've got a bit in there which is this, that's this. Then we have another protein that might fold up differently because it's missing that green part, because it's a different protein. These different versions of the same gene are called isoforms. So this might be isoform A, this might be isoform B. So maybe sometimes you get A, sometimes you get B, sometimes you get both, sometimes you get neither. So it sort of increases the number of different things you can get out of the same gene. And so another possibility, another type of alternative processing is alternative Let's see, what do you call this? It's alternative poly A tail addition. Let's see. Yes, so they call it alternative 3' cleavage. And so you can have a sequence that has 2 of these. Might have another one down here. And in some cases, we might cleave it here and give us a bunch of A's. And sometimes we might cleave it here and give us a bunch of A's. And so those are just 2 possibilities. There are other possibilities as well. And then who knows what effect that would have. It might affect the length of the protein. It might affect the stability of the RNA. It might affect all kinds of things. Summarize the different types of processing. We've already done that. Do it quickly. Splicing, poly A tail, 5' cap. Basic structure of ribosomes. The main idea is that there's RNA plus, so there's ribosomal RNA plus ribosomal proteins. And there's a small subunit. And give myself a little room over here. There's a large subunit. Now, usually these are drawn like this, with the small subunit kind of looking like that. And together we get the small subunit, and I might as well throw 3 on there in a large subunit. And then we're going to have an E site and a P site and an A site, and that'll come into play when we look at translation. And so the large subunit kind of looks like this. And the large subunit and the small subunit, they sort of get made in the nucleus, in the nucleolus, and then they come out separately into the cytoplasm. And then when it becomes time to translate an RNA, they get together on the RNA. The types of RNAs that are in these things, you can look this up in a table. And so let's see. See if we have enough room here. So we've got prokaryotes, and we got eukaryotes, and we got small subunit, and we got a large subunit. And let's see, I got to look this up. The bacterial subunit is 30S and 50S, so 30S and 50S, and then together these are 70S. You know, they don't add up, but S is kind of a strange, not strange, but kind of a unique method for measuring the size of things. And when you put the large subunit and the small subunit together, they kind of change shape a little bit, so they come out a little bit differently in terms of the size. And then you have a 40S and a 60S in eukaryotes, and these come out to be 80S. I won't bother telling you. It has to do with density. It's this technique where, I don't know, it's too long to explain. There's a bunch of proteins, and in bacteria, the RNAs are the 23S and the 5S. So these are RNAs. And in the That's in the large. And in the small, let's see, what does the small have? The small has, small has the 16S RNA. And then eukaryotes, we have 28S, 5.8S, so we got a little bonus RNA here, and a 5S RNA in the small subunit, and then an 18S RNA in the large subunit. And the S just stands for size. It actually stands for something called a Svedberg unit, but we don't really care about that. And I'm not going to ever ask you to list all of these things. What I want you to know is that basically the basic structure is the same. They both kind of look like they both have a small subunit and a large subunit. And the individual components of each of those are similar, but the specific sizes of them are a little bit different. So I think that's good enough. There's no reason to memorize that. You can always look it up, but you should know that RNA plays an important role in the ribosome. That's one of the key things. All right, now what are these things? Now we got some other kinds of RNAs. We got small interfering RNAs. We've got microRNAs. And we've got piRNAs. And these are RNAs that their basic function is to regulate the expression of other things and other genes. Let's not talk too much about piRNA. One thing we'll say is that this regulates transposable elements. So transposable elements are these things that can jump around. They're hunks of DNA that can jump around in the genome, and you don't want them jumping around too much, because if you have DNA jumping all over the place, it's going to jump into other genes, and it's going to make mutations, and it's going to cause a lot of instability in the genome. So we don't want that. So piRNA has evolved to regulate and keep these transposable elements under control. Now siRNAs and microRNAs are both similar. They both start out as double-stranded RNA, and then they get processed where they get chopped up into smaller RNAs, and then when they're smaller RNAs, then they inhibit the expression of other genes. So let's say here's another gene that has, this is an mRNA. One of these RNAs, if it matches here, it'll stick to it. And it'll cause this to get degraded. Or sometimes it'll stick to it and it will inhibit translation. And so that's kind of what they have in common. Usually, so this is an mRNA. Usually if it matches up and it causes this pathway, that's usually the siRNA pathway. And if it binds and it blocks translation, that's usually the miRNA pathway. So miRNAs block translation, siRNAs cause degradation of the RNA. Let's see. That general process of interfering with the expression of this RNA is called RNA interference. Or it's often abbreviated RNAi. And so a lot of times that's actually useful as a genetic tool or a genetic technique if you want to inhibit the expression of a gene, of a target gene. You have a gene that's getting expressed at a high level, and you want to block it for some kind of a maybe as a gene therapy thing or something like that. You take double-stranded RNA and you put it into the cell. That gets chopped up into smaller bits, and then it can bind onto these target RNAs and prevent them from making proteins. So they're very similar. They both silence gene expression, use slightly different mechanisms. Both start out as double-stranded RNAs, and these double-stranded RNAs are 21 to 22 nucleotides. And these double-stranded nucleotides, there's a couple of proteins involved. So it goes through a big complex called the RNA induced silencing complex, or RISC, and one of the proteins in there is called dicer, and what dicer does is it chops up the RNAs, chops them up into these smaller bits. So these guys. So dicer does this, and then these proteins, when they go, or these RNAs, when they go and find their target, they're carried to their target by a protein called argonaute. Write that over here. So this protein called argonaute carries, so dicer chops up the RNA into smaller pieces, and then argonaute carries it to its target. And the main difference is siRNAs cause it to be chewed up, miRNAs cause it to be causes it to block translation. And then the other difference is matches the target perfectly. siRNAs match the target perfectly, and miRNAs match less perfectly. And then cause mRNA degradation. And these guys block translation. And I think that's about it. Differences. Yeah, that's close enough. Alright, how are miRNAs processed? Kind of talked about that a little bit already. Let's see. Alright, so basically, this isn't all that important. It's made as a longer double-stranded RNA like that. And then after dicer, it gets cleaved into a smaller thing, and then eventually it gets cleaved into these single-stranded things. So it goes through a single-stranded sort of hairpin loop first. And I would just recommend you look at the book to look at the details of how that gets spliced. If you know that dicer chews up, if you know that this occurs in the context of risk, and so this ends up being in the risk RNA-induced silencing complex and argonaut, and then one of these protein, this gets bound by argonaut, and it then goes to target genes for translation inhibition. Duchenne muscular dystrophy is caused by a mutation in a gene that comprises 2.5 million nucleotides and specifies a protein called dystrophin. However, less than 1% of the gene actually encodes the amino acids in the dystrophin protein. On the basis of what you know about all this stuff, provide a possible explanation for the large size of the dystrophin gene. When they say provide an explanation, I mean, we don't know why it's that big. So what it's really asking is, how can the gene be so huge and only 1% of it be used? And so the basic answer is that it's huge, and there's tons. Well, we don't know how many. I mean, we do know, but I don't know off the top of my head how many exons there are. The answer is they have a lot of really big introns, and so then all of those introns get removed, and you get this, and you make a protein. And so that's the point. But I like to use this example just to give you an idea of what a real gene looks like. So 2.5 million nucleotides. That's a lot. So that's 1% of the entire genome, just about. Maybe 0.1%. So it's a really, really big gene. But only 1% of that gene codes for the protein. So that means that 1% of the gene is made up of exons, and the rest of it is introns, or 5' UTR, or 3' UTR, or other things that don't get translated into protein. So it just gives you an idea of the scale of that. These introns are not just some sort of random, unusual thing. They're really important parts of genes, and there are actually genes where the vast majority of the gene is introns. For the ovalbumin gene shown in figure 4.3, I'm going to go find what 14.3 looks like. So I'm going to the ebook on my other computer. Getting high tech on you. Alright. I'm having a hard time getting to figure 14.1, figure 14.2. This thing's kind of slow sometimes. Alright, figure 14.3 shows the ovalbumin gene, and it shows it in two circumstances. So ovalbumin is just a protein that's put into eggs, and there are okay, so hold on a second. Yeah, it's just so the ovalbumin gene has 8 exons. So there's an exon here, 2, 3, 4, 5, 6, 7, and then kind of 8. And so each of these is a little bit bigger than this. So these are the exons of the gene. So if I do that the way I've been drawing it, I would draw it like this. So we got 8 exons. And then it transcribes and puts all those things together. Transcribes and splices and all that. Alright, so this is a lot of big long question here. Just to build up to the question, where would the 5' untranslated region be? Well, we don't know. They don't tell us where the start codon is. So we know that this is 5' untranslated region. And depending on exactly where the start codon is, the start codon could be in the second exon. But we're just going to assume usually it's in the first exon. So this bit here is going to be our first exon. And this part is the 5' UTR. And same thing here, we don't know exactly where the 3' UTR is because we don't know where the stop codon is. But it would be a reasonably safe bet to say that the 3' UTR is in the last exon. Certainly part of it is in the last exon, but we could have a start codon here and a stop codon there. And then anything to the right of the stop codon is the 3' UTR. Anything to the left of the start codon is a 5' UTR. But I don't think they show us exactly where it is. So, but it's just kind of getting you to think about where the genes are, and then they're just showing this. The other part of that figure shows a different gene. How do mRNAs of bacterial cells and the pre-mRNAs of eukaryotic cells differ? How do the mature mRNAs of bacterial and eukaryotic cells differ? Well, bacterial cell, so these are bacteria. There's no cap. Well, this is pre-mRNA, so this is pre. And this is pre of the eukaryotes. So they look kind of the same at this point. This is the pre-mRNA. So they're just RNA. And then the mature mRNA, the bacterial one looks the same. The bacterial one doesn't change. There's no processing after transcription, so it just stays the same. And then each of these gets codes for its own protein. Gets translated and makes its own protein. And then eukaryotes, we've got our introns spliced together. We got a poly A tail, and we got a 5' cap. Can't imagine there's much more to it than that. Let's see. The other thing that's different, and I don't have a lot of room here, is in bacteria, if you've got your DNA and you've got RNA polymerase, and we're making an RNA, even before that RNA is done being made, a ribosome hops on and starts translating. So in bacteria, we have, well, I wonder why that turned into a straight line. We have what we call coupled transcription and translation. So coupled transcription and translation. Transcription and translation happen at the same time in the same place. In eukaryotes, we don't have translation yet because this guy has to leave the nucleus and go out to the cytoplasm before it can do translation. The 5' untranslated regions encoded by sequences in the promoter, exon, or intron of the gene. Exon. Because it's not the promoter. So the promoter never even makes it into the RNA. So that's important. The transcription start site is here, and that's where transcription starts. And this exon is going to contain the start codon, and everything to the left of it is the 5' UTR. I think we've done this one a million times. Do I have to do it again? So a typical eukaryotic gene, pre-mRNA and mRNA derived from it. Alright, gene. Believe me, if we were having an exam on paper, this is always a question. And then splice these guys together. And then where are these things? So we've got, so these are exons. Where'd these guys go? Hey, come back. Alright. So these exons are put back together. Poly A tails down here. Caps here. 3' UTR. Promoter. AAU, AAA consensus. And that's also here. So this is here as well. It's part of the 3' UTR. Transcription start site is here. 3' untranslated region, we got it. Intron. Poly A tail. Alright, so I think we got it. How would the deletion of the Shine-Delgado sequence affect bacterial mRNA? Here's your RNA. Here's your SD. If we deleted that, so it's no longer there, the function of the Shine-Delgado sequence is for the ribosome to bind and recognize it, so we would get no protein, or very little protein. You might get some random translation. But Shine-Delgado sequence, the function of the Shine-Delgado sequence is to initiate translation, or to allow the ribosome to bind, and without it you don't get it. What would be the effect of moving this sequence 10 nucleotides upstream? So let's say we've got one here. And then we're going to have another one, and we're going to move it up this way a little bit. So upstream is going to be to the left, or to the 5' region. Alright, so I don't know. Depends. What number is this? 26? I gotta look this one up. Well, I mean, it says so this is going to cause cleavage and poly A addition signal there, and then this would shift this site, and so then we'd get an RNA like this, and we'd get an RNA like this. So we might get an RNA that's 10 nucleotides shorter. Maybe. I don't know if it's precisely that spatially oriented, or if there's another sequence down there. It depends what the sequence is. Alright, so here we'd get no poly A tail. Here we obviously have no poly A tail. The main function of this poly A tail is to stabilize the RNA, so we'd get degraded RNA. If we don't have a 5' cap, we'd get less or no translation. It's just kind of a way to get at what the function of these things are. So that's about it. This, again, depends where this is, but if we get some mutation in the middle of this intron, it's not going to have much of an effect once we translate and splice this out. If we get rid of that intron, we get rid of that mutation, and this should still be fine. So we should have no effect. However, if we hit the specific branch point A that's involved in splicing, or if we hit some other sequences that are necessary for splicing, then we might not be able to splice the intron out, and that could affect the protein sequence. So here we'd get no poly A tail, so we'd get a less stable RNA, and so we'd get less protein out of it, because the RNA would get degraded before it could get translated. Yeah, it's kind of the same thing. I'm getting tired of answering that question over and over again. Alright, what's this? A geneticist isolates a gene that contains 8 exons, then isolates the mRNA, makes the DNA single-stranded. He mixes the single-stranded DNA and RNA. Some of the single-stranded DNA hybridizes with the complementary. So now we have a gene. So this is DNA. And he's got 8 exons. So that's DNA. And then here's the RNA. If we hybridize this together, what hybridize means we're going to stick the two things together, and wherever there's a match, it's going to stick together. But if there's not a match, if there's an exon, it's not going to have a corresponding region in the RNA to stick to, and so that will loop out. So what you'll end up with is the RNA and then the exon part of the DNA sticking to it, and then the intron part of the DNA looping out. And it's the DNA part that loops out because it's the DNA part that has the extra sequences in it. And this is how they originally discovered introns and exons. So it would look kind of like that. Obviously it wouldn't be all lined up and look perfectly like that on the electron microscope, but that's what they're getting at. Geneticist discovers two different proteins encoded by the same gene. One has 56 amino acids, the other 82. Provide a possible explanation for how the same gene can encode both proteins. Lots of different possible explanations. Let's go with a simple one. Let's say we got our normal gene, and sometimes it gets spliced like that, and then this makes a protein of 82 amino acids. Same gene, maybe a different cell type. Now we're going to splice and skip one of those things. These are exons. Now just 1 and 3 are together, and maybe this one makes 56 amino acids. Lots of different ways, but that's just kind of an easy way to do it. Figure 14.12, often called a 12. What conclusion can you make about the relative sizes of the two proteins produced by alternative splicing in figure 14.12? Yikes. Alright, so we'll come back to that one. Alright, we've got this thing going on again. Alright, so alternative splicing takes place in more than 90% of human genes. Researchers have found that how a pre-RNA is spliced is affected by the precursor's promoter sequence. I thought I deleted this question. Don't worry about that one. In fact, I don't think we have to worry about any more of these. Especially since I don't know how the heck that happens. It's just some kind of random thing going on. So let's call it. It's been over an hour. That's good enough. If you have any questions about RNA, just let me know. And really have a solid understanding of gene structure, 5' UTR, all that kind of stuff. So, if you have any questions, let me know.